This recording is for Biology 1407. This is the third chapter in Unit 4 on plant phylogeny, structure, and physiology. Chapter 30. So, plant organ systems. This applies primarily to angiosperms. In this unit, we're really looking at the structure and function of angiosperms. A lot of this also applies to gymnosperms like pine trees, and to the other vascular plants as well, in their own specific ways. So, we formally recognize 4 different organs in a plant. The roots, the stems, the leaves, and the reproductive structures, whatever they may be. So in this particular case, we're looking at a dandelion right here, and it has a taproot, which is an underground stem with lateral roots coming off of it. We have a very short above-ground stem. It's the nature, the growth pattern of this particular plant, with an inflorescence of leaves coming out of it. And then a continuation of the stem right here supporting a reproductive structure, which is modified leaves. Every part of a flower is really a modified leaf. Every part of a cone on a pine cone is a modified leaf from those plants. So these are modified leaves up here making up, in this case, the flower, or perhaps the mature seed head of a dandelion. It's hard to tell what the artist intended right here. All right. There are 3 types of tissue in vascular plants. Epidermal tissue, which is an outer covering. And then within that, ground tissue, which fills out the bulk of a plant. This contributes the bulk of the contents of a herbaceous plant, of a non-woody plant. And then scattered about inside that ground tissue is going to be vascular tissue, which consists of xylem, a system of dead conductive cells that passively transport water and ions, dissolved salts, that is to say, generally speaking, vertically through the plant from the roots and evaporating out of the leaves. Evaporation is the power source. And then phloem, which is a connected set of living cells that move metabolites, organic materials, from one part of the plant to another, generally from where they're produced to where they're consumed. So the epidermal tissue surrounding the plant consists of at least one layer of cells covering the surface of stems and leaves. And that will also be present in below-ground portions of the plant and is involved in water and ion uptake. The epidermis on the above-ground parts of most plants is going to be covered by a waxy cuticle, so the epidermal cells themselves secrete that waxy cuticle. And then there's going to be openings in that cuticle and openings in the epidermis for gas exchange. We'll see about that in a moment. The below-ground organs in the roots, at least the portions of roots actively involved in absorption of water, are not going to be coated in a cuticle. It won't have that waxy covering. Some terminology for the ground tissues of a plant, and like I said earlier, this is the primary composition of an herbaceous or green plant, a non-woody plant. And those ground tissue cells are involved in all of the really active metabolic functions of a plant, storage of metabolites, and also contributing to structure of the plant. We'll see about that in a moment. Some terms that you see within a leaf, the ground tissue is usually referred to as mesophyll, so that's the middle of a leaf. Phyll is the Greek prefix for a leaf, and meso means middle. In a stem, the bits inside the ring of vascular tissue, or just towards the center in general, will be referred to as the pith, oftentimes dead. It was living when the plant was very tiny, but as it's gotten bigger and more mature, the pith dies out a little bit. And then cortical ground tissue, which just means the outer part of a cylindrical structure. And this generally means outside of the vascular tissue. In the root, we also are going to have a cortex and some sort of pith structure within there as well. All right. When we look at a stem, some terminology that hopefully you're already familiar with. So you can divide any stem into a node and an internode. So a node is the point where we have branching, whatever that may be. Internodes are the spaces between points of branching. All right, so node, internode model is the nature of all stems of all plants. Now, the length of those nodes, the length of the internodes, the frequency of the nodes, details can vary terrifically right here, but all plants retain these two features. And when you're looking at a stem, next important thing is that you're going to have an apical bud. The tip of the stem contains an apical meristem, and so that allows for the stem to continue growing and extending. The tip of the stem is called the shoot. We can also have axillary buds, which are lateral. They're along the length of the stem. So, in animals, axillary is an anatomical term referring to where the limbs, break away from the body. So our underarm is an axillary region in our anatomy. So the axillary buds are points, where we can have a leaf branching off of a stem, or where a small branch is branching off of a bigger branch, or whatever you might have. But lateral branchings are led by an axillary bud. Then finally, a modified portion of a stalk that can extend away from a stem supports a leaf, and that is called a petiole. So that is the long cylindrical stem of a leaf. All right, so here we're looking at a very typical herbaceous dicot. You can tell it's a dicot because the leaves are branched, and you can see some hint of the branching venation in there. And here's a node right here with 1, 2, 3, looks like 4 leaves coming off of it in this particular case. Some older leaves, a pair of older leaves, and a pair of younger leaves. And then there's the long internode section, and then another node where we have leaves branching off. When we get to a flower, assumably up at the top on this plant right here, what we'll have is nodes and internodes that make up the pieces of the flower as well. And you can imagine the same sort of structure down on the roots. So here, if we take a look at a stem, this is a cross-section that has been stained, and we're looking at it under a microscope. We've got a scale bar right here. So all the way across here to about here is about 1 millimeter, about 1,000 micrometers. Over here is about 2 millimeters, 2,000 micrometers. So it's about 2.5 millimeters wide, this little micrograph of a very small stem. And what we're looking at is the 3 types of tissue that make up the stem. We've got an epidermis around the outside right here. How do I know it's the epidermis? Well, it's on the outside. It's the boundary. That's the function and the location of epidermis, via boundary. It'll be creating a waxy cuticle that covers the outside of these outer cells. Then inside that, the whole thing's filled with ground tissue, which is all the stuff that's really mostly in blue here. That's all ground tissue. In this particular case, lots and lots of cortex around the outside right here, and then some pithy material towards the inside right here. But also, because it's a very young stem, the stem is dominated by vascular tissue, which is most of the business that we're looking at inside here. And so here it's illustrating this is a vascular bundle, which consists of xylem tissue towards the inside. Xylem always has these really large diameter, relatively large diameter conductive cells, and then phloem tissue to the outside right here, more phloem out here. And this, you can tell it's phloem if you look carefully in some places, because we're going to have a large cell, small cell, large cell, small cell sort of motif, which is typical of phloem. All right. A little bit about ground tissue. So ground tissue comes in 3 different forms, and you need to know this. There are 3 different types of ground tissue, and these are in increasing order of specialization. The most general ground tissue is called parenchyma. A slightly more specialized ground tissue is called collenchyma, and then the most specialized variant is called sclerenchyma right here. So PCS, parenchyma, collenchyma, sclerenchyma. And what's the difference? Well, parenchymal cells have a very small, thin, relatively flexible cell wall, Oftentimes the cells are cross-connected to each other. They're not particularly specialized in shape, but they do lots and lots of work. They're metabolically very active, so most of the business of staying alive is done by parenchymal cells in a plant. Collenchymal cells are still metabolically active cells. In a lot of ways, they're like parenchyma, but they have thicker cell walls, and so they provide structural support. The cell walls are substantially thicker, but the cells are still alive, metabolically active, and can be cross-connected to each other to kind of work as a megatissue. Finally, the most specialized form of ground tissue is called sclerenchymal cells, and sclere, when you see this prefix right here, it means thick, or like like a callus is a sclerophilous structure, or scleroderma structure on us. Sclera means thickened. So sclerenchyma contains these much heavier cell walls, including what's called a secondary cell wall inside of the primary cell wall, so closer to the center of the cell, or inside the outer wall. That usually has some structural proteins in it, like this is where we'll see lignin in plants that produce lignin, and that makes for a very hard cell, which makes for a very hard structure, whatever those cells are made out of. Those cells at maturity are dead. To produce a secondary cell wall basically kills the cell, so at maturity it's a dead hollow shell. All right. And we'll see this, for instance, the hard outer shell of a peach pit. What makes it really hard is super tough sclerenchymal cells. All right. Those sclerenchymal cells can come in two forms: fibrous, long ones that'll reinforce the stem of a herbaceous plant. If we're searching for fiber in plants like cotton fiber, or the fiber from yucca, or the fiber from hemp, and all of those plants, that is going to be the result of these long, slender, fibrous sclerenchymal cells. That's what we're after when we harvest the fiber from those plants. And then sclerids, my peach pit outer wall that I was referring to earlier, is composed of these sclerids right here. All right. Here's a cross-section of the stem of, as it says right here, St. John's wort, Hypericium perforatum. And here we have pith in the center, a ring of vascular tissue. This is going to be the xylem with the very large diameter hollow cells right here. Phloem outside of that. And then some cortex ground tissue out here. So this is going to be parenchyma and or collenchyma. This is going to be mostly parenchyma, maybe a little bit of collenchyma. Collenchyma is the ones with the slightly thicker cell walls, if you can see them. Same thing out here. And then some dermal tissue around the outside right here. And if we could look in close enough at the outer edge of the dermis, we'd see there is a waxy surface on there, a cuticle. All right, so here we're taking a more magnified view of some cortex of a stem, and here is some collenchymal tissue. And notice the very thin cell walls, very thin cell walls. All right, dermal tissue. Again, that's the outer surface of a plant, of any of the herbaceous parts of a plant. So green stems, young roots, leaves, flowers, petals, all of those structures are all going to have a dermal layer on them. And within those dermal cells, we'll have pairs of cells called guard cells that can move away from each other, can create an opening between them called a stomata, is the proper name for that pore. The stoma, or stomata plural, one stoma, is the opening between two guard cells. When they close up tight, the stoma is gone or closed. When they inflate with liquid, they actually open up, and that causes the stoma to be open. Another type of cell that we'll have on dermal tissue besides these guard cells is cells called trichomes. So these are cells that produce a hair that sticks up off of the surface of the epidermis. So if you've ever seen a fuzzy leaf, you're looking at a whole bunch of trichomes on the surface of the epidermis of that fuzzy leaf. These also produce the root hairs. A variation of the trichome is the root hair on roots, which is the actual fuzzy sort of surrounding of the tip of a root, the very end of a root, and this creates a whole bunch of surface area for the absorption of water and nutrients. The trichomes will be covered in a waxy cuticle. The guard cells will have a waxy cuticle over the cell portions, not the stoma for obvious reasons. That's a hole. The root hairs won't have a waxy cuticle because their whole purpose is to absorb water and nutrients from the soil. So here's some imagery. We're looking at the surface, dermal surface of a plant. And these will be some dermal or epidermal cells right here, and this is going to be a pair of guard cells right here, creating this stoma right here. Here's more of an illustration, 2 guard cells right here. And it's the way their cytoskeleton and their cell wall is organized. When they inflate with liquid, when they become highly pressurized with liquid, they actually flex into this open position right here and open the stoma. When the plant becomes dehydrated, the guard cells become dehydrated. When they become dehydrated, they become flaccid, and they actually fold shut and close off the stomata, limiting the amount of water that can evaporate from the inside of the leaf, because the outside of the leaf is covered with wax and is it doesn't allow water to evaporate. So that's the closed stoma versus the open stoma. So the function of the stoma is to regulate gas exchange, to allow for gas exchange, and to regulate, by closing, to regulate the amount of water lost by the plant from evaporation or evapotranspiration. And interestingly, they close passively when they lose water pressure. They open only when there's high water pressure inside the tissues of the plant. Vascular tissue consists of both xylem, which is this heavily reinforced dead at maturity tissue that moves water. The water that's evaporating out of the stomata is coming from the xylem system. And then a series of cross-linked, highly permeable living cells makes up the phloem. So dead tissue at maturity, living tissue at maturity for the xylem and the phloem. Water and dissolved minerals, sugars, amino acids, hormones, signal molecules, all of that sort of stuff being moved in the phloem. This is more equivalent to our blood, if you will. And in both cases, we have xylem and phloem distributed through the plant in the form of a vascular bundle. The strands are called vascular bundles, and they always have xylem and phloem together. They always come together as a pair, running the length of the plant. As I said earlier, if you look at a dicot, one of the ways of knowing you're looking at a dicot is if you cut the stem and you look at it in cross-section, you'll see that the vascular bundles are arranged in this very characteristic symmetrical ring. So here's a cross-section of a monocot stem. This could be corn, this could be a grass, this could be a palm tree, this could be anything, and there are some that will be hollow. So like if this was bamboo, it'd have a big hollow hole in the center right here, but the rest of the stem would look like this. And then here's a dicot, and consistently for the dicot, we've got the vascular bundles in these very neat, well-organized rings, vascular bundles in a ring right here. We're going to have cortical ground tissue in the inside, which is going to be pith, and ground tissue around the outside here, which is called cortex. Cortex just means location. So this outer layer is the cortex. This inner layer here is the pith. And our vascular bundles consist of xylem and phloem, and then in this case, these reinforcing fibrous sclerenchyma cells right here that provide structural support, particularly protection from being penetrated from the outside. Think of a sucking insect sticking its proboscis in here. It's going after the phloem right here, which is full of sugar. It wants to stick its proboscis in there and drink this liquid. This fibrous, tough sclerenchyma protects that as a kind of an armored cap. When we look at the vascular bundles of the monocot stem right here, they're not as heavily protected, but they have the same arrangement. There'll be xylem towards the inside, the larger diameter cells, phloem towards the outside, and some number of sclerenchyma cells surrounding this vascular bundle, keeping, kind of protecting and giving it structural rigor. When we look at xylem tissue, we want to identify two types of tissues that we find in there, two types of cells. The more primitive and more general xylem tissue is tracheids. Remember tracheophytes? Tracheophytes are plants that can produce tracheids. So the tracheids are the generic sort of xylem tissue that we find in almost all plants. They are fairly small in diameter, very long, narrow cells with an end wall, and the walls have these holes, which can be seen under a microscope, allowing connection from one cell to the next. In some dicots, but really highly present in monocots, grasses will have vessel elements, which are much larger diameter hollow cells, open at the end that, collectively a whole pile of vessel elements joined together makes what looks like a big hollow straw running through the inside. Big, under a microscope, small to the naked eye, but a hollow straw passing through the stem of the plant. These allow for much more rapid movement of large amounts of water. These are smaller and less, able for water to move through. These are more resistant to damage. These are more easily damaged. Phloem tissue also comes as two types of cells. There are two that you need to know about, but this is a different relationship. There are the cells that conduct liquid from one place to another. These are called sieve tube cells. And remember that phloem consists of living cells. So these are living cells that are cross-connected to each other, very much like tracheids. Anatomically, they look a lot like tracheids, except these things are dead at maturity. So no cell membrane, no organelles, just a hollow thing, just a hollow cell wall. But these things are alive. So there's a cell membrane internally, there's organelles, there's cytoplasm in the cells. But we have lots and lots of very large plasmodesmata connecting one cell to another through these, so a perforated sieve plate is this structure on these sieve tube cells that connects one cell to the next. The sieve plate is characterized by lots and lots of very large cell-to-cell connections. In plants, those are called plasmodesmata, and they allow the cytoplasm to flow continuously through the sieve tube cells. Now, since the cells do have a cell membrane, but they don't have any other internal organelles, including nuclei, they have to be maintained and kept alive by a specialty cell called a companion cell that will be found adjacent to each sieve tube cell. So every sieve tube cell gets its own companion cell that keeps it alive. And these cells have all of the ribosomes, the endoplasmic reticulum, the nucleus, all the stuff that the sieve tube cell has gotten rid of so that it can be a specialized cell just for transporting liquid. All right. Let me just see something here. Yeah, okay. So here we're looking at stems. This is the transition from a herbaceous stem to a woody stem. So dicots are capable of producing wood. Not all of them can, but those that do. And so a young tree will be herbaceous. It will have a green stem like this, and the very tips of the shoots of the branches will also be herbaceous. They'll have a green living epithelium on them, cortex, pith, ground tissue, vascular bundles arranged in a ring. And what happens is the vascular bundles fuse together to make a cylinder, a vascular cylinder. And that vascular cylinder it has stem cells right in the middle here between the xylem and the phloem that continuously produce xylem, which is going to be left behind on the inside, and continuously produce phloem, which will be pushed outwards towards the outside. And so here's the first year's phloem, here's a second year's growth of phloem, here's a first year's xylem, here's a second year's xylem, and we would create a third year and a fourth year and a fifth year. And what happens is the entire stem keeps getting larger and larger in diameter as that vascular tissue keeps secondarily growing through these secondary meristematic tissue here. And the first year, all the older year phloem dies and becomes crushed and non-living. Only the most recent phloem is functional, just like only the most recent xylem is still used to conduct liquid. And the dead phloem plus sacrificial epidermal tissue that produces a material, the dead material called cork, piles up and forms collectively the bark of the plant. And as this cylinder keeps getting larger and larger in diameter, the bark characteristically splits. And that's the split pattern that we're so familiar with on the trunk of a tree. All right, roots. So remember, dicots have a tap root with branches coming off of them. Monocots have a bushy pile of all equal roots. So here's a taproot system. That's what we're looking at up here. And here's a fiber root system. I think in this photo they're implying that this plant has a fibrous root system, but there is a taproot somewhere going off laterally. It's not going to go deep below the water table right here because roots can drown. They like to get near water, but that's where they stay. They don't go deep down into the water, below the water table in soil. But we've got an eroded riverbank right here where we can see the root system of this tree. All right, so root tissue, again, on the outside, it's going to have an epidermis. It's going to have an epidermal layer right here. This typically will not produce wax because the whole point is to absorb water from the environment. Extensions of those epidermal cells will be the root hairs. These fibrous extensions of single cells that increase surface area and improve their ability to absorb water. So here's a cross-sectional drawing, or a longitudinal section, I'm sorry, of a root tip. And we've got the sacrificial callus of dead cells on the outside here called a root cap. Right here is going to be the apical meristem, the stem cells that keep dividing to produce all the other tissues of the stem in this case, and then leaving behind them ultimately an epidermis, ground tissue cortex, and a vascular cylinder that consists of xylem and phloem. And then here we've got these root hairs growing out from the individual epidermal cells right here, extending the surface area of the root. Here's a cross-section of a dicot root. And so here's the vascular cylinder at the center. There's going to be xylem in the very center, phloem towards the outside. And that is always the conserved arrangement of vascular tissue. Xylem always closer to the center. Even if it's a vascular bundle out here, the xylem would be closer to the center, the phloem would be closer to the outer surface. So here we've got phloem around the outside of this vascular cylinder. And then something called the endodermis, which is a layer of cells that have a waxy cuticle between them, and that ensure that water that normally moves between cells, all of this pathway here is permeable to water, but when water gets to the endodermis right here, all of the liquid must be moving across the living membrane of a cell, and so that allows the membrane to regulate which ions are brought across. And so the water in the cortex of the root right here has dissolved in it whatever's dissolved in the water out here in the soil, including toxins, heavy metals, all kinds of poisons, whatever. The living endodermal cells right here with wax in between them ensure that only things that they permitted across their own living membranes get into the root bundle right here and get transferred to the rest of the plant up above. That's the role of the endodermis right here. So we have an epidermis that does not produce wax, but we have an endodermis that produces wax between the cells, not on their faces. The cells are still permeable to water via osmosis, permeable to ions by selective, ATP-powered movement or hydrogen ion-powered movement of things across the cell membrane. And so there's selective absorption at this point. Another ring of cells just inside the endodermis is called the pericycle, and it's usually fairly evident under a microscope. Extra thick cell walls right here. All right. That endodermis, the layer of wax between each cell, is called a Casparian strip. And again, it ensures that water and solutes only move across living membranes of the endodermal cells instead of slipping between the cells, which is what happens everywhere else in a plant. The only place where it can't happen is crossing this endodermis. So we can see here, this is a great way for plants to exclude toxic compounds and pathogens, keeping them out of the vascular tissues of the plant. When we look at a monocot versus a dicot, there's some ways to tell the difference pretty quickly. In a monocot, the vascular tissue in the root is arranged in a ring that's kind of reminiscent of the stem of a dicot. This is a stem of a dicot, but this is the root of a monocot, and then a very, very thick cortical region around outside here, but a ring of vascular tissue. And oftentimes this pith will be hollow. There'll be an airway through here. Grasses, think of grasses with that hollow center. but that's a root. This is a root. In dicots, inevitably the vascular bundle in the center has this sort of X-shaped anatomy in here. We've got an X-shaped structure with a whole lot of xylem in it, most of the xylem being towards the middle of the X, and then pockets of phloem in quadrants, in 4 different quadrants around it right here. Then a whole lot of cortex again, and then the epidermis. And the boundary out here of this outer layer is the endodermis, with its waxy cuticle, not cuticle, but waxy layers between the cells, forming a regulated transition between the free-for-all of the cortex and the free-for-all of the inside of the vascular cylinder. So as you can see right here, these are all roots we eat. So roots can be modified pretty substantially. In plants, these are inevitably to store foods for later use, like over a winter, and then shoot back up the next year. That's the classic with bulbs, like these onion bulbs right here. Or these corms, like this corm, C-O-R-M, like these beets down here, or these radishes right here, or various other things. Sweet potatoes, any number of different modified roots. Okay. We can also have some interesting modifications of roots up above ground. These are banyan trees and fig trees over on the side right here. Both are characterized by these stupendous secondary roots which prop the plant up. This is a real typical adaptation to either living in water, like in a very, very loose, sort of silty, muddy bottom, or on very thin soil over rocks. So we've got this extensive surface area of load-bearing structure right here to hold the plant up. All right, let's talk about leaves for a little bit. A leaf consists of a stem, which is called a petiole, and then a blade called the lamina. Think of laminating. When you're laminating a sheet of something, that's what a leaf is like. Think of a laminated piece of paper with plastic on both sides. The plastic on both sides might be the epidermis with a waxy coating on it, and then the paper on the inside would be the mesophyll of the leaf, and the whole thing together would be called a lamina. Anyway, so the lamina and the petiole, all right. Some terminology: leaves that are attached directly to the plant without a petiole are called sessile leaves. There's quite a few plants that look like that, but more common is to have the petiole and then the lamina of the leaf some distance away from the stem. If you look closely at where leaves come off of plants, you usually find a second little kind of green appendage, and that is called a stipule right there. A lot of times the stipule can be modified into quite a complicated structure. All right. Most leaves are going to have this midrib, and then if it's a dicot, we'll have branches from that midrib. If it's a monocot, we'll have parallel strands of vascular tissue parallel to the midrib. So this must be a dicot because it has branches. Here's a primary branch, secondary branch. I'm not seeing any tertiary branches on this, but they very well can occur. All right. Here's a monocotyledonous leaf with the parallel venation, an iris or a lily. Here's an oak leaf. Here's a ginkgo biloba right here, which is a branched, what's called, where do they have it right here? Dichotomous venation. Dichotomous means branching again. So this is branching. And notice we've got these branches in the veins, branches, branches, branches. Lots of little branches. So it looks like a fan, but it's also the result of branching. This is the result of very obvious branching. This is not. So we can see lots of different versions of leaves. In case you're not familiar with it, there are simple leaves, which is one petiole with one leaf, or a petiole that branches into multiple lobed leaves, palmately compound right here. Pinnately compound, which means compound like a feather is, with things coming off the side of the petiole right here. Sorry, the petiole goes to the center here, and then this leaf, this is the central midrib of the leaf with a whole bunch of secondary leaves coming off of it. And then here's a doubly compound. This is characteristic of Fabaceae. Think of a mesquite tree. Its leaves are going to look like this right here. And that's called doubly compound right here because there's a petiole leading to a compound leaf. Each compound leaf, this is a compound leaf. It has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 leaflets on it. And then each leaflet is divided up into its own little micro leaflets as well. So that's what we mean by doubly compound. Take a look at a pecan tree. They're all in bloom and in leaf now outside, and you'll see that it's a compound leaf like this with typically 11-fold symmetry. So it'll be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 leaves, unlike this one, which has 7-fold symmetry. All right, when we take a look at a leaf, we want to know a little bit about its anatomy. So remember, generally speaking, a leaf, like every other organ of a plant, has an epidermis filled with ground tissue. That's this stuff here. And then scattered about inside that ground tissue will be vascular bundles. That's true for every organ everywhere. Epidermis, pile of ground tissue. Suspended within the ground tissue will be vascular bundles. In the case of a leaf, the ground tissue is formally referred to as mesophyll, which consists of these photosynthetic cells arranged in kind of parallel columns. That's the palisade here. These are going to be parenchymal cells. The type of cell is, a thin cell wall, very metabolically active, really complex cells. That's the palisade mesophyll. And then a spongy mesophyll, which is a layer of more irregular cells that have lots of air spaces in between them that allow air to percolate through the leaf, which we need for gas exchange. Or as it says right here, in the case of aquatic plants, that spongy mesophyll is a sealed sponge full of air that makes the leaf less dense than water so the leaf can float. So here is the epidermis with a waxy cuticle on top of it. The epidermis of the underside of the leaf with a waxy cuticle on it, a pair of guard cells creating a stoma for gas exchange right here. Spongy mesophyll with lots of room for gases to move around in here. Palisade mesophyll. The only thing we haven't talked about is the vascular tissue. Let's see if we can find that. Well, it's implicit in here. So somewhere nearby will be a vascular bundle with xylem towards the top and phloem towards the bottom. And if you think about how the leaf appears to fold off of a stem, the upper side of the leaf, if it's clasped, folded up against a stem, the upper side of the leaf will be touching the stem of the leaf, and the xylem therefore will be closer to the center of the leaf, while the underside of the leaf will be exposed to the outside. It'll be on the outside if the leaf is folded up tight against the stem, like before it unfurled, and the xylem will be, facing towards the outside of the stem that way. So that's how that makes sense. Xylem always towards the top of the leaf, phloem towards the underside of the leaf. Always, always, always. Here's some examples of modified leaves. So, these are still leaves, just like flowers are still leaves, but they're highly modified for a second function. So they've given up most of their photosynthetic activity. There's still obviously chlorophyll at work on, mesophyll cells right here. So it's still photosynthetic, but it's just not super good at it. The red is to attract certain things that can see in color. A lot of insects are, color vision insects, and the red is attractive to them. Also, birds are attracted to red. And this is a Venus flytrap. Everybody knows about that. Here's a pitcher plant, which has these waxy hairs, trichomes, on the epidermis of the inside of this tube right here that face downwards. So something walking on here kind of gets forced to go downwards, and the hairs become increasingly slippery, and it falls into a pit of liquid right here, which is full of lytic enzymes that break polymers apart. And so the insect will be slowly digested in here. Glandular tissue on the inside of the pads here of this Venus flytrap will secrete digestive enzymes as well, those lytic enzymes, just like the pitcher plant will right here, and digest that insect. In both cases here, the plants are eating insects, quote unquote. They're really dissolving insects, and they're going to absorb the nitrogen that is released from their amino acids. They're after the nitrogen in the amino acids of the proteins of the flies. That's what these plants are primarily after. To a lesser degree, they'll be after phosphorus and sulfur and other elements, but it's particularly nitrogen. The reason plants, so to speak, eat insects and any other animal that they can get to die on them or in them or around them is to get the nitrogen that's contained in the proteins of the plant. Nitrogen is the hardest nutrient for plants to extract from their environment. All right, water potential. Is signified by this Greek letter right here, psi. PSI is psi. Remember, Greek pronunciation, the second consonant is the one we say, so this is psi, like psionic, like psychedelic. And it's symbolized by this little, what looks like a trident right here, all right? And that is the symbol for water potential. And that's usually measured in something called a megapascal. It's a unit of pressure, right? Or pressure right here. And this will look complicated, and it is a little bit, but hopefully you can make sense of it. The total water pressure, the water potential, which is the potential of water to move from one side of a membrane to another, or to move from one location to another, is the result, is called the, this is the water potential of the system. This could be the atmosphere, this could be the xylem, this could be the cytoplasm of the cell, whatever, soil. That's the system we're talking about. And it is the total, it's the sum of all the water potentials, which equals the water potential of solutes, plus the water potential of pressure, plus the water potential generated due to the force of gravity, plus the water potential due to the adhesion of water to a hydrophilic surface. S, P, G, and M, respectively, right here. All right, and so we add all of those together. So if they're all positive, this number is going to be really big. If two of these are negative and two are positive, we're going to end up with a smaller, intermediate number. And if the two values that are negative are equal to the two values that are positive, we'll end up with a water potential total of 0. So the arithmetic is very straightforward. Okay, so how does this work? Well, the most important one for plants, and the one that they can manipulate the most, is the water potential due to solutes in water. And you know that if you're familiar with osmosis, you're familiar with that phenomenon. And so let's go ahead and look at that for a second. Here's an illustration right here. This is an experimental setup where we've got a tube of liquid with a semi-permeable membrane right here that allows water to pass across it, much like a cell membrane, but doesn't allow sugar molecules or salt molecules, whatever, salt ions, whatever it may be that we want to dissolve in here. So this is very standard experimental setup. So in this particular case, we can take a tube that initially looks like this, dissolve some sugar on one side of the tube. The sugar can't pass across this membrane, and so now the concentration of water is lower here than it is here, because here it's pure water, but here it's a mixture of water and sugar, and so water moves down its concentration gradient via osmosis across the membrane right here and piles up on this side of the tube. So that is essentially what all living cells do to absorb water from their environment. They make sure that the contents of the cell are more concentrated with sugars and salts and proteins than the environment they're in, and then they'll preferentially pull water across the cell membrane into them via osmosis. Now this experiment here allows us to illustrate another thing as well. If you set this experiment up here, water will rise up here until it hits an equilibrium point where it won't go up any further. Add more sugar to the water right here and it'll rise up a little bit more until it gets to an equilibrium. The equilibrium is going to be the downward force of gravity pulling on this bigger column of water, exerting a pressure from right to left across this membrane right here, counteracting the osmotic flow from left to right. And so we'll reach an equilibrium. And that would be an illustration of the interaction of the potential due to solutes and the potential due to gravity, for instance. All right. All right. Here's another thing. If I set up this system right here and I increase pressure here, I put a plunger in here and just push down on this liquid, I can push water across the semi-permeable membrane here, all right? I could do it over here. I could apply pressure here and push water across this membrane. I can apply pressure to the sugary water right here, but what I get is pure water accumulating over on this side of the tube right here if I apply a positive pressure right here. And that's what reverse osmosis is, if you've ever heard of it. That's how we can make fresh water by pressing salty, contaminated water across a membrane that's porous only to water molecules. The holes are too small for anything else. And so we can create fresh water. It takes a lot of energy because it takes a lot of force to press water in reverse osmosis across a membrane, but that's, what we do. Osmosis is water being drawn towards solutes. Reverse osmosis is filtering solutes out of liquid by pushing water in the opposite direction of osmosis. Reverse osmosis. That's how most ships make fresh water while they're at sea. If we apply a vacuum, negative pressure, we can suck water across this membrane and pull it up the tube here. So some examples of how you can manipulate water potential, and plants do that. They manipulate the solute potential, and they can create positive and negative pressure to draw water in different directions. Remember, no living cell can grab a water molecule and actually move it. What they can do is move solutes, and by accumulating solutes, removing solutes, they can cause water to be drawn from one compartment to another. All right, so because water is drawn in osmotically into a cell, that cell wants to expand, but as it expands, the membrane presses against the cell wall, which is quite strong, and this creates something called turgor pressure. So plants inevitably are inflated by turgor pressure. That's what makes a freshly watered plant stand up nice and tall, and as it becomes desiccated due to evaporation, it loses turgor pressure and the plant can start wilting and collapsing. That's what happens to your vegetables in the fridge over time. And here's some values. Water pressure can be equal to as high as 1.5 megapascals. That's the metric unit of pressure right here. And as you can see right here, that's equal to about 200 psi. You inflate your car tires typically to around 35 psi. So this is up to 6 times the pressure of your car tires being inserted on the inside of a cell wall of a plant. So the key way in which plants can move the movement of water, manipulate, the pressure of water, is to change the solute concentration in that water. Wilting plants, turgid plants. Wilting on the left, turgid on the right. How does water move through plants? Well, water is moving through the xylem system due to negative pressure due to evaporation. All right, so there is a negative pressure. Water is actually being sucked through the xylem system. As you can see here, -2 megapascals. That's going to be around -300 psi. So there's a fairly extreme tension on the water in the xylem system, pulling water up through a plant. And that movement of water is called transpiration, and it's powered by evaporation. So the long name is evapotranspiration. All right. So here we're looking, and here is evapotranspiration up through the roots, up the stem system, out the stems of the branches, out the stems of the twigs, all the way out the petioles of the leaves, into the mesophyll of the leaves, where we've got vascular tissue right here, and we have little openings that allow water to evaporate out of the vascular tissue, out through the stoma, and as that water evaporates, it creates a negative suction on the water inside the vascular tissue right here. So the water is literally sucked up the plant by evaporation, powered by sunlight. The red arrows refer to the water kind of acting as a chain right here, adhering. The water molecules adhere to each other, so if one is lost, another one is pulled along. And the water also adheres to the inside of the xylem system. It's basically the same thing paper towels are made out of, and you know how paper towels attract water. So water is pulled up a little bit by capillary action due to adhesion. And where is that water coming from? The soil. And this can appear to be awfully dry soil, but the suction, the potential to pull water via the system is so powerful that plants can pull water out of what appears to be very bone-dry soil and rehydrate themselves. So the water is being pulled up and lost to the atmosphere. Now, obviously, this will work really well when the atmosphere is very dry. When the atmosphere is really, really wet, the movement of water through this system is very sluggish. High humidity, sluggish movement. Low humidity in the atmosphere, very rapid movement. Super dry soil, slowed down, sluggish movement. Really, really wet soil, rapid movement. So that process is passive. It's driven by evaporation, powered by heat, which is coming from the sun. The movement of liquid through the phloem system is active. This is a pressure system. So here are our sieve tube cells, the sieve plates connecting them, a whole bunch of plasmodesmata. Here's the companion cells that keep our sieve tube cells alive, the companion cells. So that's phloem, the tissue called phloem. It consists of these two types of cells. And there's also cross connections. These cells cross connect laterally to companion cells near them and to other tissues around them. So this, this is not totally in isolation here. We've got cross connections to other tissue. And the phloem system and the xylem system work in concert. So the xylem system provides fresh water to the entire plant as it's evaporating out through the leaves that can be used and drawn into the phloem system. So here, for instance, we've got some leaves. This would be palisade or spongy mesophyll of a leaf, where we're doing a lot of photosynthesis, so we're producing a whole bunch of sugar molecules. The red circles represent sugar molecules in the form of sucrose, the disaccharide in this case. And that is actively pumped into these phloem cells by ATP-powered pumps or ATP-powered proton pumps, and then the hydrogen ion concentration gradient is used to drive the sucrose pumps. So secondary pumping. ATP powers the pumping of hydrogen ions. Hydrogen ions going down their concentration gradient powers the sucrose pumps. That's the way it really works in plants. And so the sucrose accumulates in this phloem tissue, creating a high osmotic potential, so water is drawn in via osmosis. Where does it come from? Surrounding tissues. Where does that get replaced from? Xylem. And then as the fluid pressure increases in here because of osmosis in, that drives the liquid away from its point of origin. So the phloem flows away from wherever sugar is being loaded into it, carrying sugar with it. And then at, tissues that are growing leaf, growing tip, growing shoot, growing fruit, growing roots, growing anything, they're using sugars, lowering the water potential in these tissues right here, and so water tends to leave this the phloem system where there are nice big openings like these sieve tube, like the plasmodesmata. The sugars can move along with the water into these source roots right here, and that ensures more sugar is delivered to these tissues continuously. This is under pressure. Pop a hole in this tube right here, and liquid jets out. This is under suction. Pop a hole in this thing, and air rushes in to create an air bubble. This is under suction. This is under pressure, and the two work together in a collaborative transport scheme that has this sort of cross-linking of the two systems. Next, we want to look at some hormonal responses of plants to their environment. Some responses to light right here, as you can see, is photomorphogenesis. That means growth, or a change in growth based on light, as opposed to photoperiodism, which is a response to the appearance of light or the absence of light. That can be daily photoperiodism, flowers opening during the day and closing at night, or leaves tracking the sun over the day. Or it can be photoperiodism over a longer period of time, the difference between, winter, short days, and long days. And then phototropism, which is the directional growth in response to light, as opposed to a different sort of response to light right here, which is, you said right here, this photomorphogenesis allows the plant to optimize its shape to absorb light. So, puts bigger leaves where there's lots of light, smaller leaves where there's less light, no leaves where there's no light, lots of leaves where there's a lot of light, things like that. Phototropism is the actual act of growing towards or away from light. So the stem crawling along the ground until it hits a patch of light and then growing straight up would be an example of phototropism. To respond to light, plants must have a molecule that responds to light, that detects light and initiates via some sort of transduction pathway a response inside the plant. And those are collectively referred to as phytochromes. And chromes are simply molecules that interact with light, oftentimes have colors, and phyto means plant. So phytochromes typically have two versions right here. We can see phytochrome red and phytochrome far red. R means red and FR means far red. Red is 667 nanometers around that wavelength, while far red, so getting over into the infrared range, is about 730 nanometers. So phytochrome is a molecule that, when it's exposed to this wavelength of light right here, converts into one form, and when it's exposed to this wavelength of light right here, converts into another form. And the phytochromes are used to regulate all of these processes right here to some degree, as well as things like germination and, seasonal responses. We have, if they were getting towards the end of the year, and we'll find that our we're getting more and more red light right here. We might trigger, the beginning of the conversion of leaves from their red to their senescent, from green, their mature green, to their senescent red coloration, like the turning of leaves in fall. Germination of of seeds in the ground is oftentimes controlled by detecting wavelengths of light right here. All right, so let's look at the mechanism a little bit. You do need to know this one mechanism. And so the phytochrome molecule has these two stages. When it's in the active state, it's phytochrome in the it's in the active state right here, active phytochrome. Now, when it's exposed to far red light right here, that inactivates the phytochrome, converts it into this inactive form. In its active form, it is transduced across the cell membrane and induces the cell to do something. All right. So if we hide the plant from light in darkness right here, the phytochrome goes from this active state right here to this inactive state right here, where it's sensitive to the presence of red light. If it's exposed to red light, it'll switch back to the active state right here. And the way this works is, if a plant is exposed to red light, that means that it's exposed to sunlight, direct sunlight coming from the sun. And what we want is the plant to grow in that direction and make itself exposed to that red light. Remember, chlorophyll can absorb blue and red light, so red light is good, good for making a plant grow. So red light is a stimulus for growth of the plant. If a plant is exposed to only far red light, that means it's in the shade. Far red light is the infrared light that makes it through a leaf. It's the red light that's not absorbed by chlorophyll. It's too long wavelength, and it actually makes it down into the lower reaches under leaves. And that tells the plant it's in the shade. So it's not going to produce leaves right here. The cellular response is going to be to maybe allow stem growth to continue, to have the stem keep growing until it hits light, and then the stem stops growing and leaves start growing when it hits light. There could be a lot of different responses right here, but the idea is to get what's going to have leaves in the presence of red light, because red light means real light coming from the sun. If they're only exposed to far red light where we don't want the leaves to be, the plant can't photosynthesize. So it's evading far red light and looking for red light. There's also sensitivity to blue light. And those are phototropins right here, and these are proteins that switch between two states, active and inactive. Inactive when they're not exposed to blue light, and active when they're exposed to blue light. And they are another mechanism to induce the plant to bend its way towards light, all right? And to bend away, from light also exists. That's negative phototropism. Roots will do negative phototropism. Stems and leaves will generally do positive phototropism. Leaf opening and closing, chloroplast movement, stomatal opening are all regulated by blue light detectors. In this case, these bluettes right here, the azure bluettes, which is kind of an oxymoron, one word describing the other word, are photoperiodic. They, show a daily, phototropic response where they bend towards the light. They track the sun as it's bending over them, as it's going over them by bending, so they're always facing the light. Time lapses of plants are really illuminating because they're so much more mobile than we normally think of them being. Gravitropism is the ability to detect gravity and grow in response to the force of gravity. A stem exhibits negative gravitropism; it grows away from gravity, and a root expresses positive gravitropism; it grows towards light. And the way it works is that we've got cells in the cortex of the roots and some cells in the cortex of stems that have these starch, the vacuoles full of starch, which is really, really dense stuff. And so what happens is the vacuoles fall against the edge of the cell containing them. And that causes one side of the cell to release growth hormone or a stimulating hormone, typically auxin. The hormone is auxin, that stimulates let's see, how does it work? Oh, sorry. Releases a hormone that inhibits the release of auxin, inhibiting growth on this side, while we do release auxin over here, and it stimulates growth on this side of the root right here, and so the root bends down to track these things. When the signal is sort of equal, left and right, the root will grow straight down. And so that is gravity detectors using negative gravitropism, and the actual sensor are these heavy plastids full of starch inside the cell walls of some specialized cortical cells right near the growing tip of the leaf, right near the apical meristem. And if you want to know about the molecules, I'm not going to ask you to remember them. Here is the signal pathway right here. And it is a surface membrane receptor system with water-soluble signal molecules and all of this sort of stuff interacting with each other. And this is an illustration of the transduction pathway. Y'all don't have to memorize this, but if you're curious, you can read about these abbreviations and get a short lesson in signal transduction. Another thing is plants are obviously sensitive to touch. Venus flytrap capturing flies is a pretty obvious description of the sensitive, positive thigmotropism, movement towards a stimulus. Here's another example of positive thigmotropism. A lot of vines quest around, and when the tip of the vine touches something, it preferentially grows in a way that allows it to curl up and tighten about the object it hits, and that's how vines can grab a hold of a structure and grow up that structure. Those are both examples of positive thigmotropism. Negative thigmotropism would be things like a root, when it hits a rock, growing away from that rock until there's no rock under it, and then it can grow down again. So that's how they go around obstacles. Lots of stems normally, if the stem hits something because it's being blown around by the wind, that causes negative thigmotropism, and the stem will tend to bend in its growth to bend away from the thing that it's hitting. But that's an opposite to the way climbing vines work, where they grow towards whatever they hit. These 5 plant hormones you need to know about. You need to know their names and what they do. So there's a grid of 5 hormones right here. There's quite a few more out there. There's lots more we could study. We'll just keep it down to these 5 hormones and these 6 sorts of responses that might be possibilities. And notice they're somewhat, exclusive to each other. All right, gibberellin stimulates germination and development of fruits. All right. Stimulates production of flowers, basically maturation. So, a seed, gets going because it's heading towards maturity. A plant grows until it takes on its mature form and starts producing reproductive structures, and then the development of those reproductive structures. So gibberellin induces all of these things. Commercial grape growers spray their grapevines with gibberellin, and that makes the grape bundles produce more grapes and bigger grapes closer together. So, that drives maturation of the grapevine heads, flowering of the grapes, and development of those fruit. Auxin is a hormone that drives stem elongation. So, the growth of, development of mature structures in a stem. Also the development of a flower at the tip of a stem and maturation of fruit. So auxin has a number of different functions there having to do with growing of the tips of the plant, and flowers are characteristic of the tips of the plant. Cytokinins. Cytokinins, this means cell movement. Cytokinin, cell movement. These induce cells to divide, which contributes towards growth and maturity, flowering, and fruit development, in all cases by driving cell division. Auxin drives cell growth, a cell getting bigger, like in the G1 stage of the cell cycle, or the G2 stage of the cell cycle. That's what auxin primarily stimulates, is physical growth of a given cell. Cytokinins stimulate division of cells. ethylene is a gas. This gaseous hormone is what's responsible for the brown bag effect, where if you put a pile of fruit or any kind of vegetable matter in a brown bag, but particularly fruits, bananas being the most vivid example, the ethylene gas starts accumulating in that bag, and if you smell this distinctly sweet, gaseous, rich sort of smell. Well, that's ethylene, and it stimulates fruits to mature and flowers to finish their process and go on into producing fruits. So flowers will age quickly, and produce seeds and produce fruit, and fruit will mature and become, mature. Ethylene gas also causes leaves to let go of trees. So abscission is the act of leaves letting go from a tree. When they drop their leaves, that is abscission. Abscission. And ethylene stimulates leaf abscission, which once you've had your flowers and produced your fruit, oftentimes that's the next thing the plant's going to do, is get rid of its leaves as it prepares for its dormant season in a temperate environment or in a very dry environment, if it's a wet, dry sort of, tropical, semi-tropical environment. So those are the roles of some of the roles of ethylene, and it's the one molecule. These are all distributed in the phloem system of the plant. Ethylene is a gas, so it's released into the atmosphere. This also allows trees to communicate with each other. This acts like a pheromone, inducing coordination of reproductive events in all the members of a given species of plant by coordinated release of ethylene gas. So trees coordinate their flowering and fruiting. Abscisic acid, pretty obviously, is going to induce abscission. So this is another hormone that's released into the phloem system of the plant, and that induces leaf abscission right here. And also seed dormancy. So as long as there's lots of abscisic acid in a seed, it keeps the cells in the seed from mitosing and developing and becoming active. It causes cells that are sensitive to abscisic acid in the base of the petiole of a leaf to die, preferentially die, causing the entire leaf to fall off the tree. That's the function of abscisic acid. Now, the way it works in seeds is if you soak the seed in water for a long time, the abscisic acid concentration in the seed goes down because it's water soluble, it's an acid, it dissolves in water, and so the concentration goes down in the seed, and when the concentration goes low enough, the seed can now germinate. That's why you soak your beans, to get rid of the abscisic acid. So know these 5 hormones and their effects. None of them, influence all 6 of these, phenomena: germination, growth, flowering, fruit development, abscission of leaves and stems, seed dormancy. But each, influences a few of them, and these are organized kind of in descending order, moving from germination to seed dormancy as we work our way down the table here. And that concludes this recording, the 3rd chapter of Unit 4 in Plant Structure and Function and Evolution.